Hi, everyone. Good to see you this morning. My name is Gaurav Shah. I am the CEO of Rocket Pharma, and very excited to spend about 10 to 12 minutes telling the story as fast as I can. And I love Q&A, so would love to entertain whatever questions come up. So I'm not going to belabor the point about what gene therapy is for this audience, obviously, but I think one of the exciting things about Rocket for me is that we've become a multi-platform company. We have indications in both the ex vivo lentiviral-based platform as well as the in vivo AAV platform, all of which are in the clinic or about to enter the clinic this year. The programs are Fanconi anemia, leukocyte adhesion deficiency 1, or LAD1, pyruvate kinase deficiency, or PKD, and infantile malignant osteopetrosis, or IMO. Those are the Lenti programs. The AAV program is our most recent addition to the pipeline. It's called Danon disease, and it's potentially the first curative therapy for a monogenic cardiomyopathy. We have several near and medium-term milestones, which I'll go through in a little detail as we move through each program. But the bottom line of starting this company and building this company is that we were building it on the building blocks of academic experts, scientists, and other peers and companies who have gone before us and already have come a long way in perfecting the ex vivo lenti technology as well as the AAV technologies. So it's like we're starting on a stage, not from the bottom, and we're going after diseases where we can be first and only in class. That's important for us. And where a little protein can go a long way, where a little protein can potentially correct the full spectrum of the disorder, of the disease. And also where clinical endpoints can be achieved in a reasonable time frame. So as you can imagine, finding assets like that is not easy. And our team has taken a few years to figure out what those assets are, bring them into Rocket, and try to develop them as fast as possible for the sake of patients. Our team draws along those lines from folks. These are folks who have done drug development, gotten several drugs approved. I'm an oncologist myself, previously at Sloan Kettering, spent a few years at Novartis, helped on Kymriah, which is the first foray of gene-modified cell therapies and the first dialogue of that kind with FDA and EMA. Likewise, our chief medical officer, our chief operating officer who's here, and others have taken something like 15 to 20 drugs to the clinic and to commercialization. So we're building a company based on people who have already learned how to integrate and build a sustainable organization. So our biggest program in terms of market size is called Danon disease. This is a devastating multisystemic disorder. It's X-linked, and it primarily affects the heart. So most people with this disease manifest with heart failure early in life. It's X-linked, so boys are affected. It's X-linked dominant, actually, so boys are affected more than girls are. Boys typically start getting heart failure in their teenage years, and the median age of survival without transplant, or even with transplant, is around the age of 20 years for boys. It's in the 30s for girls, although many girls are starting to exhibit symptoms earlier in life as we're learning real time. Heart transplant can be curative, but is associated with something like a 50% 10-year mortality rate. And of course, the heart failure does not address the other manifestations of this disease, which include, at times, profound skeletal muscle weakness, CNS changes, and others as well. Now, the US plus EU prevalence for this disease is something like 15 to 30,000. And that's estimated based on a pretty comprehensive and validated literature review. The capsid we've chosen via a licensing partnership with REGENXBIO is AAV9. And the reason for choosing AAV9 is that there is essentially a hand-to-glove fit between that capsid and the organs that are affected in this disease. So AAV9, we know, has high tropism for liver, which is affected. But other than liver, the next highest organ is cardiac, and then skeletal muscle and CNS. These are the organs affected in Danon disease, and hence we chose AAV9 to work with first. So LAMP2 itself is a protein that is involved in autophagy. Autophagy is like the vacuum cleaner of a cell. The cell creates debris, mitochondria, DNA, other proteins that need to be cleaned up, and autophagy is the process by which these particles are vacuumed up. Without a vacuum, the cell sort of expands, the vacuoles sort of build up, and in the heart, which is striated muscle, the cells can't contract and generate contractile activity, which is why these patients have heart failure. The same happens in skeletal muscle and CNS and elsewhere. This could be a case where a little protein, a little LAMP2 protein, can really go a long way. In our early preclinical work, we're showing that even at moderate doses of AAV9, and what I mean by moderate is, of course, subject to interpretation in this field, but even at 5e13 VG per kilogram, we're starting to see marked improvements in both contractility and relaxation. And by the time we're above 1e14, we've already hit the plateau of the curve there. Similarly, there's a survival benefit in mice who are treated with a moderate dose of the gene therapy, which is AAV9 based, versus those who are not treated, just the knockout mice. This is also seen across vector copy numbers, RNA, protein, and function. So there's a dose response across a trend across all those different assays. And I'm not going to go too much into it. I think this is the money slide. On the left side, you can see wild type, heart, liver, and skeletal muscle tissue. In the second panel is the knockout, where there are numerous vacuoles that build up. And the next three panels show really complete restoration of histology. Now, we have two mouse models that have validated these results. One is a young model, which represents perhaps what a younger boy would experience. And we've shown prevention of onset of histology, histologic dysfunction, and other assays. And in an older mouse model, we've seen reversal. So this could be a preventative therapy as well as potentially a curative restorative therapy. So in terms of our mouse and wild type non-human primate tox studies, we've seen relatively a benign safety profile, and we haven't seen any untoward effects in the first week or two that have been seen in other AAV programs to date. So we are now in the clinic. We have a trial up on clinicaltrials.gov. The IND was just cleared recently, and we anticipate treating patients starting in second quarter at UCSD. This will be a phase 1 program, and more to come on that soon. So that's Danon disease. Moving forward to Fanconi anemia, and I'll just highlight this one amongst the Lenti programs because it's in the clinic. Fanconi anemia is a disarray of DNA repair in which stem cells that don't have an intact DNA repair pathway fizzle out over time. So these are patients who, by the age of 10, no longer have a functional bone marrow. So the primary manifestation of Fanconi anemia is anemia and aplastic anemia. Patients can be treated with an allogeneic transplant. However, it's associated with GVHD and also 30 plus or more increased risk of head and neck cancers associated with GVHD for those who are transplanted. So while there is a potentially curative therapy, it's toxic, and even with that, the 1-year mortality is 10 to 20%. So there's a high unmet need here for something different. What we discovered in Fanconi anemia, which is unlike all the other ex vivo Lenti disorders, is that you don't need conditioning, right? So for the traditional Lenti approach, there are 3 factors that you basically have to get right, and if we can get those right, we have a chance at potentially curing these bone marrow-derived disorders. The 3 factors are a high vector copy number, a high cell dose, and the right conditioning regimen. Again and again, partners, academics, investigators, companies who have followed this formula have done very well in 9 or 10 disorders such as beta thalassemia, ALD, MLD, etc. For the first time, we're showing that at least in one of these diseases, you don't need conditioning at all because the marrow conditions itself. There is a subset of Fanconi anemia in which even a single cell, a disease cell, reverts to normal because of a second mutation that normalizes the first mutation. And that patient, over time, can actually be self-cured of Fanconi anemia, of the hematologic manifestations of it. That's called somatic mosaicism, shown here on the left panel. And this opens up the door for us to do gene therapy in Fanconi anemia with 0 conditioning whatsoever, and as early in life as possible as a preventative therapy. So it's changed the game completely in this field. And 4 out of 4 patients who were treated at CMAT, which is one of our partners in Spain, when treated with this gene therapy, the lentiviral-based product, when treated with this with zero conditioning, all 4, when followed over a year, have engrafted peripherally and also in their bone marrow. So the vector copy numbers peripherally have continued to rise up to 30 months. And likewise, the bone marrow is now resistant to alkylating agents, which is the hallmark of diagnosis of Fanconi. So the patient on the left, 2002, has 70% bone marrow resistant to alkylating agent, means 70% of that patient's bone marrow is normal. If you compare this bone marrow with the normal person, it would be hard to differentiate from a normal person. So essentially, we think that these patients have a high likelihood of long-term cure from the hematologic aspects of disease. And 2 words in the last minute and a half on LAD1. LAD1 is a devastating disorder which unfortunately leads to death in 2/3 of children by the age of 2 because of a neutrophil defect. CD18 on the surface of neutrophils is missing, so not allowing these neutrophils to extravasate into sites of infection. This is truly a case, and I'll end with this one because it highlights the essence of why we do what we do in gene therapy. A little CD18 can go a long way. Patients who have less than 2% CD18 almost uniformly die in their single-digit years. Those who have 4% or more, an increase from 2 to 4%, almost uniformly live into adulthood. So what we've done here at Rocket, and alongside many of our peers in the industry, is figured out how to maximize those vector copy numbers, those cell doses, optimize those conditioning regimens, so that we can actually pretend enough protein to these patients to bring them over the bridge here. And potentially this could be a curative therapy. We're now actually in a phase 1/2 registration trial by agreement with FDA that is starting shortly as well. PKD is the indication that perhaps many folks have heard about through the excellent work of Agios in the last several years. We're going to be targeting a more severe post-splenectomy transfusion-dependent version of the disorder via a traditional lentiviral-based approach. And in this coming year, we anticipate starting trials in Danon disease, in LAD1, in PKD. Fanconi, we anticipate having agreement with FDA as to the final registration endpoint. And we're very, very excited about all of this. And thank you very much for listening. I'm happy to take questions in the last 2 minutes, if any. There's got to be one. Call names otherwise. Patrick, I was going to call your name.
Yeah, sure. So you just presented some data at the ACC conference. Are there available take-homes that you might want to provide?
Yeah, the ACC data. So most of the stuff, most of the graphs and the pictures from ACC, we've already shown, we've already disclosed. Histology, functional correction, some of this I just showed. The vector copy numbers were interesting in skeletal muscle versus cardiac. I think that was the take-home that we've never presented before. The steep part of the curve for skeletal muscle AAV9 therapy seems to start somewhere above 1E14, which is why likely you need doses at 1E14 or higher for a skeletal muscle-based therapy. Same is true of CNS, as we've seen with many of our legendary colleagues at Avexis and elsewhere. For heart, the steep part of the curve seems to start much lower, somewhere around 3E13 or so. So even at potentially moderate doses on the lower end of the spectrum, we could see transduction of the heart in a way that you don't see of skeletal muscle and CNS. I think that was the take-home that was exciting for us. As a field, not just going into Danon, but all of cardiac-related diseases with gene therapy moving forward. Yeah, so there are, if Jonathan Schwartz was here, he knows all the references, but there's a paper by Murray from 2014, by Kacak, K-A-C-A-K from 2006, I think, and Myers in 2015. These are all on our website. Pacak, P-A-C-A-K. All of those show that both mouse and non-human primates have something like 1 log higher vector copy numbers in the heart versus skeletal muscle. RNA has been somewhat more variable. Protein has also been significantly higher in heart versus skeletal muscle. So in our non-human primate work that we're just completing, and I've said this publicly, but we have not published this, but I've disclosed this publicly. We've seen that cardiac versus skeletal muscle or CNS, which is about the same, there's about a 1 log higher vector copy number in the heart versus the others. So that's the idea, and that's the data we're going into with for our clinical trial, and we'll see what happens there. So thank you very much.